On that note, I'm going to hand over to my colleague Hugh Laverty.
So good afternoon, everyone, and thank you for joining us for today's webinar. I sort my wires out here. So today's webinar will cover all aspects of the call topic. So Claire will basically run through the call topic, covering objectives of the project, the need for the public-private collaborative research in this area, the structure of the project, expected contribution of the applicants, the contribution, importantly, of what the industry consortium are going to bring, and then what we are expecting this eventual project to deliver. What we will not cover today are the rules and procedures that cover IMI2 calls. A webinar on the rules and procedures will take place on the 9th of January 2017, and you can register for that webinar using the link shown on this slide. So before we get into the meat of the call topic, I just want to give you a little bit of orientation about IMI. Many of you are probably well aware that we are a public-private partnership, but I'm still always amazed that when I go out and I give presentations, some people aren't quite aware of what IMI is trying to do or how we operate. So I have a few slides to run across some of the main themes. That's not in detail, but you'll get a lot more detail in the rules and procedures webinar that will be held in January. IMI is a public-private partnership between the European Union and the pharmaceutical industry in the form of FPA. We were set up under FP7 as IMI1, and we were renewed under Horizon 2020 as IMI2. As I said before, we're a public-private partnership with the two partners of industry and the European Union. They are equal partners and they equally contribute. So our total budget is just over €5 billion for the program, with 50% coming from the European Union and 50% coming from FPA. It's very important to remember that the money that comes from the European Union is available for IMI beneficiaries that have been identified through open competitive call process and selected to work on IMI projects, and none of the public money goes to industry. What industry does, industry brings its own in-kind contribution to the project, so they contribute people, they contribute their own resources within each of IMI's projects. Our mission is to facilitate open collaboration and research, to advance the development of and accelerate patient access to personalized medicines for the health and well-being of all, especially in areas of unmet medical need. IMI provides a platform to set up novel and unique consortia that work on key challenges in pharmaceutical research and development in the delivery of healthcare. And as this platform, we allow engagement in a cross-sector multidisciplinary way, where the consortia are undertaking work at the forefront of cutting-edge research. Our project provides the necessary scale for combining funding, expertise, knowledge, skills, and resources. And the collaborative nature of IMI is built on trust, creativity, and innovative and critical thinking. It's also very important that by bringing together these multidisciplinary, multi-stakeholder consortia, that we are willing to learn from each other, and the different partners are able to bring new knowledge, skills, ways of working. And IMI is an opportunity to take part in transformative research that will make a difference in drug development and ultimately patients' lives. Within that, we at the IMI office and the IMI platform, we try to be neutral, where all involved in drug development can engage in open collaboration on shared challenges. Very quickly, to orientate those of you who aren't used to IMI about how our calls work, our topics are normally defined by industry. So the different industry partners come together and they identify areas where they are willing to collaborate, and they identify the resource that they're willing to bring to a call topic. They prepare a call topic text, and then we launch the call based upon those topics. At the first stage of a two-stage process, which is our normal project lifecycle, is two-stage process, we invite applicant consortia to submit short proposals. And now these consortia can have any combination of academics, hospitals, SMEs, regulators, patient organizations. It's all very dependent on the topic and the focus of research in a topic. When we receive the short proposals, they undergo an independent evaluation with a panel of independent experts. The independent experts will score and rank the proposals, and then it's very important to remember that in order to progress to the second stage, we only take one. We only take the top ranked short proposal that has been submitted for a topic. So, and then at the second stage, we invite the applicant consortium of the successful proposal to merge with industry, so to come together, to integrate, to work together as the full proposal consortium, and the full proposal consortium prepares the full proposal. This full proposal then undergoes another round of evaluation by the same independent experts that worked on it at stage one. And we move, if everything goes successful and it passes this stage, we then move on to the grant preparation phase, where the consortium come to reach agreement on a consortium agreement, and then based upon that consortium agreement, the IMI office will sign a grant agreement and we can launch the project. As Catherine said in the introduction, please be aware that from IMI Call 10 onwards, we are in the Horizon 2020 participant portal, and so we are using this as our submission tool. For those of you who are used to IMI, we no longer will be using SOFIA. Everything will come through the Horizon 2020 participant portal, so please keep that in mind. The proposal template that you will have to complete is available on our website and also within the Horizon 2020 submission tool. There are one or two little differences for those of you who are used to other Horizon 2020 programs. Under IMI, first stage proposals, our page limit is 30 pages, so it's slightly longer than other parts of Horizon 2020. And also, we evaluate proposals based upon the 3 criteria at both stages. So we assess excellence, impact, and implementation at both the first stage and the second stage. We have shown you the evaluation criteria here. I don't intend to go through them all. You get the slides, you'll be able to read them. But please be aware that these differ slightly from the evaluation criteria that you might be used to in other parts of Horizon 2020. So they've been slightly tweaked so that they're more in line with the work that we support in IMI2. It's also very important that when you're preparing your proposal that you keep in mind that these are the criteria that the independent experts will be using under excellence, impact, and as I need to go back. And as I said before, under the third criteria of quality and efficiency of the implementation. So, as I said, please familiarize yourself with these. I don't intend to go through them today. Some tips for writing a successful proposal. Most of these are very self-evident, but we still see some mistakes been made in these areas even after 8 years of operation. Please read all the call relevant material. This is particularly important for those of you who are not used to IMI. Please understand our rules and respect them. All of the information is available on our website. We always advise potential applicants or people who are thinking about applying to IMI is to please start forming your consortium early. In our experience, this is one of the key steps in being successful, is building your consortium and starting that as early as possible. There are partner search tools and there are networking events that are available for you to start building your network outside of IMI, but this is something we can't really stress strongly enough, that the sooner you start this work, then the sooner you'll be prepared to submit an application. It may seem self-evident, but please provide reviewers with all of the information requested to allow them to evaluate your proposal. Based upon the evaluation criteria, based upon the form, everything should be there. There should be no open questions or no doubts regarding what you intend to do. Experience as well has also taught us that it is better to finalize and submit your proposal early, because I think with every call we do get requests from applicants who have had problems in submitting their proposals. And unfortunately, when these are related to the applicant or the applicant server or their firewall, there's nothing we can do about that. So we would advise you to try to finalize and submit your proposal early. And if you have questions related to the call topic, then please contact the program office. Do not contact the FPA topic writers. It's very important in our efforts for transparency and openness that all questions come to the program office. We instruct the FPA topic writers that if they are approached by any potential applicant, they should direct them directly to the program office anyway. So save yourself an email or a phone call and just come to us directly. Some common mistakes that we often see, as I've mentioned before, we still have many examples where submission deadline is missed for different reasons. And as I have said, if it is on the side of the applicant, there is nothing we can do. Also, please be aware of eligibility criteria, such as the minimum of 3 legal entities from 3 member states. If this is not met, then your proposal will not be evaluated. Other important points to remember is that our projects tend to be quite large. They tend to have quite a broad range of objectives. So you need to make sure that your proposal addresses all of the objectives of the topic. We get some very good proposals, but they may only cover 50 or 60% of the objectives of the topic, and in that case then the proposal will not be successful. It's very important that your proposal and your consortium is able to address all of the objectives as laid out in the call topic text. Also, please remember in IMI, a very important part of what we do is we are looking to impact upon the drug discovery process, drug discovery and development, and also eventually onto patients. So having a proposal that is scientifically excellent is not enough. We expect all proposals to be scientifically excellent, but they must have an impact. If we get an excellent proposal with a limited impact, it will not be successful as well. It will not be scored highly. And also keep in mind, in the spirit of IMI, you are applying to a public-private partnership. You are embarking upon wanting to work with an industry consortium, so you should already be reflecting on the complementarity with the industry consortium at the short proposal stage. And so these are some important considerations you need to keep in mind. We're often asked, how do I find project partners? Well, I think you can never start early enough or soon enough, and you should be thinking about building up your network of contacts outside of IMI anyway, because when a call comes up that you want to apply to, you're then able to tap into your already preformed network. We share the contact details of the participants that have joined the webinar today, and so that's another opportunity for you to network, because there are many people who are interested in this topic. And then, of course, there are different partner search tools. We have a partner search tool, the German National Contact Point, as a partner search tool, and Fit for Health is the wider Horizon 2020 partner search tool as well. You can also get in contact with your local IMI contact point, and their details can be found by following the link. You can also talk to your Health National Contact Point, because they may be aware of other partners that are interested in applying. And of course, we have a LinkedIn group, which is always an opportunity for you to network outside of when we have our calls. So, somebody is very desperate to join us, so we're trying to unplug the phone from the wall. So one of my final slides is on SME participation. This is something we'd really like to encourage all applicant consortia to remember. SMEs have a really important role to play in innovation and bringing new ideas in research and development. So we would encourage you to reflect upon including SMEs in the participation or in the formation of your consortia. And when I looked at this topic, because it's very much focused on research and development, I think there's lots of opportunities for specialist SMEs to participate, whether that be around providing technologies and expertise to perform the screens or the next generation sequencing, whether that be in the area of mass spectrometry, that is described within the call text, whether it's bioinformatics, or whether it be around target identification, assay development, HTS chemical probe generation. There is a lot of opportunity for SMEs to contribute to this project. And with that finishes my introduction, and I hand you back to Catherine.
Thank you very much, Hugh. And now we're going to hand straight over to Claire Stephan from Pfizer, who's going to present the core topic. Over to you, Claire.
Okay, thank you. So as Catherine said, I'm Claire Stephan. I'm from Pfizer. So thank you for taking the time to dial in to this webinar today. And we're going to be talking about unlocking the solute carrier gene family for effective therapies. Next slide, please. So what was the, when we thought about what was the key driver or motivator for us assembling this call, was the central challenge around solute carrier proteins or transporters, which I'll probably refer to as SLCs moving forward. So they're heavily understudied and underexploited as drug targets. So when you look, the SLC family comprises the largest family of membrane proteins with approximately 400 members, and as you might imagine, they play vital roles across many different cell types and in all tissues. When you look at the human genetics, 50% of SLCs are associated with a human disease-relevant phenotype compared to only 20% for the broader human genome. Thereby, this is suggesting that these SLC or this family of proteins are more have a relatively important position in disease. However, when we go to the literature, there was a recent review where the analysis looked at publication on the topic of SLCs, and they determined that they're highly understudied. So greater than 200 SLCs, or over half the family, have less than 15 publications in the public domain. So therefore, this gene family is really highly understudied. On the flip side, there are drugs out there, so they have given rise to some very important drug classes, such as the SSRIs for depression and the SGLT2 family for diabetes. However, when you look at the SLC gene family relative to other families, for instance, the olfactory GPCR, they're highly represented. So only 2 to 3% of the current drug targets are SLCs, and if I compare that to the GPCR family, 25% of the family represents drug targets. So again, in summarizing, we feel that SLCs are a highly druggable family and attractive as therapeutic targets, but they're, from both a drug discovery point of view as well as a biology, basic understanding, they're heavily understudied and underexploited as drug targets. So this is the premise of the challenge and why we want to move forward with this proposal. Next slide, please. So why do we need a public-private collaboration in order to, as we say, unlock the SLCs or the potential in this gene family? And the driver behind that is the scale of work required to unlock these is really beyond the reach of any single company or single institution. So therefore, and what we feel is necessary to unlock the family or move the field forward, it requires a range of scientific disciplines and methods, and especially if they're applied in collaboration or in concert, will really move the field forward. And specifically, some of these disciplines are techniques for the production, isolation, and characterization of integral membrane proteins, which SLCs are, metabolomic techniques, so they can be applied for more rigorous characterization of endogenous SLC substrates, or even deorphanizing. There's a good percentage of the family that what the substrate the transporter transports is unknown. Advances in methods for studying protein interactomes, and they should allow for the interaction of obligate binding partners as well as novel revealed regulatory mechanisms. So again, the regulation of SLCs are under study. New physical methods and techniques for the detection of membrane transport, and lastly, gene editing capabilities that will allow for manipulation of the SLCs to be further studied in vitro and in vivo context. Next slide, please. So we feel the pre-competitive nature of this proposal will bring together a level of funding, focus, and stakeholder commitment that should be sufficient to unlock the potential in the SLC family. What will also entail is employing a system-wide approach to the investigation of the SLC family. To date, they've been studied more at the single transporter level. It will also leverage novel techniques and methodology for understanding and profiling SLC. It will coordinate the focus on SLC transporter deorphanization and correct substrate identification. And lastly, in the spirit of the IMI, it will deliver new open access research tools, techniques, reagents, and knowledge to the biomedical research community for the study of SLC proteins. So then, next slide, please. If I move on to the objectives of the full proposal, so to unlock the SLC family for drug discovery efforts to be conducted at will across the entire family, we want to accelerate the study of the detailed study of SLC transporters. So the reagents or techniques this will entail include generation of cell lines, reagents, small molecule tools, and antibodies for the study of SLCs, development of novel methodologies for assessing transporter function, deorphanization or identification of endogenous substrates of SLC transporters, generation of purified SLC proteins for more detailed study, as well as characterization of the SLC interactome to identify obligate binding partners. So this summarizes the objectives of the full proposal. Next slide, please. So what we anticipate the expected impact to be is that this project will deliver new open access research tools, techniques, reagents, and knowledge to the biomedical research community, and this will rapidly accelerate the pace of research in the field of SLCs, and advances of this magnitude will impact both basic research and drug discovery. In terms of providing therapies, as SLCs are broadly expressed, their therapeutic potential will span many disease areas, and some to highlight include oncology, immunology, neuroscience, metabolism, and cardiovascular disease. So the impact of unlocking the family should be broad in terms of delivery of potential therapeutics. Next slide, please. So this is the suggested architecture of the project, and as I mentioned, SLCs are a large family. They comprise of approximately 400 proteins. So in part one of the proposal, we're very broad across the family, and what we want to do is generate reagents as you see outlined. So in work plan 1 would be generate cell lines that cover the entire family. Work plan 2 would develop a general deorphanization methodology that we could utilize across the family. And work plan 3 is to develop assays for the majority of SLCs. So these, the purpose of part 1 is to deliver reagents and technologies broadly across the family. And then we've also parsed into part 2 where we will generate, the consortium will generate a priority list of 72 SLCs, so a more focused list that will be put together collaboratively, and then we will study these SLCs in further detail. So work plan 5 comprises of generating high-quality protein reagents that can be applied to a variety of techniques. Work plan 6 is to develop high-throughput cell-based and cell-free assays that would enable screening of files. And then work plan 7 details would be studies and methodologies for understanding the SLC interactome. And then part 3 comprises the amalgamation of part 1 and 2, which is around knowledge, integration, management, and sharing, as well as establishing an academic collaborative network and project management. So in sum, part 1, 2, and 3 would be comprise the full proposal. Next slide, please. So if we go into some of more details on the proposed key deliverables of the full project. So as we've covered already, we aim to deliver new research tools, techniques, reagents, and knowledge to the biomedical research community, such that on completion of this 5-year project, the pace of research in the SLCs will increase, and thereby this leads to accelerated discovery of new targets and drugs which target the SLC family. And below, I will systematically list the key deliverables we've identified. So for the gene family deliverables, generation of cell systems which express in a functionally competent form a large majority, so we're targeting greater than 80% of the family. We are also targeting to generate methodologies that will deorphanize, again, 80% of the family, and enable the correct assignment of endogenous substrates to the majority of the SLCs. They transport a wide variety of substrates across the family, and then even within certain specific transporters. So to really diligently identify substrates and their respective transporters. Next slide, please. So we also would like to develop novel and broadly applicable screening methodologies for SLCs that will enable drug discovery efforts. We also want screening methods that will be, for 80% of the family, and then we would like specific assays that will cover greater than 50% of the protein family. And then additionally, SLCs are located at a variety of subcellular localizations. So we are specifically highlighting assays for monitoring SLC function for those SLCs that are localized within intracellular compartments, which has proven to be challenging to monitor function or screen for small molecules. Next slide, please. So when we get into part 2, which is more detailed study or focus on, and we have approximately 72 SLC targets in that portion of the proposal, we would like to generate purified SLC protein and/or cell-free or reconstituted systems that will facilitate the detailed study of the SLCs. Additionally, generation of high-quality biochemical reagents and techniques for the studying IMI2 SLC family members, generation of highly selective SLC antibodies. So again, this has been challenging for integral membrane proteins. Techniques to define the interactome of SLC members and to begin to build a knowledge base of understanding binding partners, regulatory mechanisms, SLC transporter protein complexes. And the last would be the deliverable of high-throughput screening assays for studying a focused set of SLCs, so that will be highly enabling to drug discovery efforts. So that covers all the key deliverables for the full project. And now on next slide, please. So the expected contributions of the applicants. So again, an advisory role with the ability to leverage expert knowledge in the SLC field, expertise and input on experimental design and reagent generation, the delivery of validated reagents such as cDNA clones, cell lines, transgenic or knockout mice models, functional genomic tools such as SI or shRNA or CRISPRs, as well as antibodies. And additionally, technical expertise, so mass spectrometry for metabolomics and proteomics studies, expertise in protein expression for integral membrane proteins, protein characterization, protein and protein ligand crystallization or X-ray structure determination, possibly, screening technologies to monitor SLC transporter function and elucidating protein interactomes. Also expected is a proven ability for the development of novel biochemical and or biophysical assays, an ability to integrate bioinformatics, so from various omics platforms that may be utilized, and then also an infrastructure to support large-scale and high-throughput experimentation, as we're looking to apply this broadly across the family. Next slide, please. So what are the expected in-kind contributions from the excuse me, industry consortium? So again, we can bring expertise on experimental design and reagent generation to the proposal. We also can provide validated reagents such as cDNA clones, cell lines, SI, or CRISPRs, antibodies, small molecule ligands, substrates, or imaging agents. Again, we can bring the same overlapping technical expertise, so mass spectrometry for metabolomics and proteomics, protein expression, protein characterization for integral membrane proteins, protein and protein ligand crystallization and X-ray structure determination, screening technologies to monitor SLC function, synthetic and computational chemistry expertise for chemical probes. We also have the ability to integrate bioinformatics from open platforms, and we also will bring advanced high-throughput screening methodologies and expertise to the proposal. So next slide, please. So hopefully you have a good feel of the landscape for the SLC family and what we hope to put forth in this proposal. So what's in it for you? So for the academic researchers, there's the opportunity to discover novel biology and deepen our understanding of SLC transporter structure or function. So as I emphasized at the beginning, these are highly understudied fields, so there's significant opportunity there. There's also the opportunity to deliver significant scientific advancement in the field of SLC transporters, and the opportunity to play a role in the identification of new therapeutic targets. So if the proposal works as outlined, will advance the field as well as advance therapeutics in this against this target class. For SMEs, there's the opportunity to develop and validate new technologies on the structure function of SLC transporters and the ability to link molecular and mechanistic insights on clinically important membrane transporters to human disease. For patient organizations, there's the potential for novel and effective therapies for a broad spectrum of diseases that have unmet medical need. based on the broad expression of this family within tissues and relevance to disease, broad range of diseases. So last slide, please. So just to highlight, as I mentioned, I'm from Pfizer, so with my colleague Dave Hepworth, we're coordinating the proposal, and we have other colleagues on the line from Bayer, Boehringer Ingelheim, Novartis, Sanofi, and V4 Pharmaceuticals. So these are the pharma partners that are we're collaborating to support this proposal. And that was my last slide.
Thank you very much, Claire, for your presentation. It's now your turn to ask questions. As I mentioned earlier, there are two ways to do that. If you want to ask your question over the phone, simply click on the raise hand icon and we'll unmute your microphone. If you want to ask a question in writing, just type the question into the question panel and click on send. I'm just going to unmute as well David Hepworth of Pfizer, who's also on the line, in case he wants to ask a question. And we have some questions in already, and they are from, one moment please. So we have the first question from Paul van Veldhoven. How can the functionality of expressed SLCs be tested for orphan carriers?
So if I hear the question, how can function be tested for orphan substrates?
So the question is, how can the functionality of expressed SLCs be tested for orphan carriers? If you want any clarification, we can maybe unmute Paul and ask him for some more clarification on that.
I can try and take a stab at it. I think that really is a key question, and one of the things that we would like, academic engagement with that problem. That's quite a common problem and a challenging problem, and I don't think I have the answer to it, but I'd say that's a big motivator for why we want to set up this research project, because techniques to get at that question will be important.
Yeah, I mean, I guess if I'm understanding the question correctly, the other thing we've thought about is, if you look, which we didn't highlight here, is potentially using a cell line. So if the transporter is endogenously expressed, we'll assume we have full functionality of the transporter, and may use functional genomics to knock out other transporters or knock out the transporter. So having a full requirement of what's required for full functionality of the transporter may be obviated if we work in an endogenous system. But again, as Dave said, this is going to be a key challenge, and that's why bringing experts together from pharma and academia. So it's a great question, and it's, I'd say, something hopefully we could tackle in the proposal. But I guess you could argue it's a chicken-egg argument. You don't have functional transporter, you don't know what it transports.
Okay, thank you. We have a question from Doron from Polygene Animal Models. And they say, we are collaborating in a few consortia circling around issues of drug discovery. Target validation in animals is always an issue. Is it here too?
Yeah, I mean, I think it'll be, so there's certain, so again, the deep study will help. So if we understand, so certainly within a certain SLC family, we've seen from drug discovery where the different isoform or subfamily members, the expression may be differential from preclinical models to humans. So one is being aware of it, so they may not be applicable. At times we'll knock in the human form. So I think compared to every other target class, the challenges would be the same using preclinical models for predicting human disease or efficacy of therapeutics.
I would say that probably the I guess in my opinion at least, the the biggest barrier to studying SLCs more broadly has been challenges of dealing with the things at the protein level. I guess once you're armed with an antibody or a small molecule, I think the in vivo challenges are probably not any significantly different to other classes. Although, as Claire said, there can be some questions of say redundancy in transporters and different transporters functioning differently in different species. But I guess you see that with all kinds of target classes.
Okay, thank you. We have a question from Julian Dow on eligibility. Will an American partner be eligible as a third country? I don't know if you want to take that one, Hugh.
So American partners can apply, but under our rules, they would not be eligible to receive funding in this topic.
Okay, thank you, Hugh. A question from John Unit, again maybe for Hugh. What are the timelines for this IMI course?
So the call we expect to be launched before the end of this year, so imminently, within the next week or so. And then we would expect a submission deadline for stage 1 at the end of March. So with 3 months from launch to submission, basically.
But as I said earlier, the indicative topic text is already online. So if I were you, I would go to the IMI website, go to the Future Topics page. There's a direct link from the homepage, and download the text and start reading and start working now, because the information that you need is there. A question from Ad Eiserman. What will the total budget be?
So the total budget is currently 24 million. So that is 12 million of in-kind contribution coming from the FPA companies. So that means there's 12 million of public funding available. So I think the key number for the public applicants is that there's 12 million available, but the total budget will be 24 million.
Thank you. We have another call procedure related question, and I really would encourage anyone who has questions about the call procedures to sign up for the webinar on that on the 9th of January, because then you really should get all the information you need. But we have a question from Edwin Kunji. Will the successful consortium be able to adopt excellent parts of other consortia for the final proposal? Hugh.
So it's an interesting question. I don't have a direct answer for that, other than no, that isn't how our evaluations work. What you would expect is you would expect the successful consortium to have submitted an excellent proposal in the first place. It may need have some small shortcomings, but it should not need major restructuring, because that's not possible under our rules. Otherwise, it wouldn't be successful. The other thing we have to keep in mind is confidentiality of applicants. So if you apply to IMI, your application is dealt with in confidence and under very strict confidentiality. So again, that prevents us from doing this sort of cherry picking of different things between different proposals. But basically, based upon our criteria of excellence, impact, and quality of implementation, you need to have an excellent proposal in the first place that does not require major changes. Otherwise, you won't be successful.
We have a question on the core topic from Winfried Neuhaus. Could also some specialized cell models, especially, for example, also disease models, for example, biological barriers, be included in the work program, or is this already too far away? For example, models of the blood-brain barrier, the kidney epithelium, and so on.
So we do not have such cell models or disease models proposed, like the blood-brain barrier. I think that would be sort of this proposal. The output should sort of lay the foundation for that type of work, but we don't have that proposed right now. I don't know how it works, if it's considered in scope or out of scope, but right now we do not have sort of complex cellular models or heterogeneous models proposed.
Okay, thank you. We have another question from Paul van Veldhoven. Are there any attempts or efforts to list up already available knockout mice or cell lines?
Sorry, read the beginning of the question again, I'm sorry.
So the question is, I don't know if it's are there or is there any attempts to or efforts to list up already available knockout mice or cell lines?
We have not to date, but I'm assuming we could. I guess we're aware of I guess within the proposal, we have not yet done that to date, but we could.
Okay. So is that within the scope of the topic then, or?
Yeah. So the goal is to deliver, you know, cell lines. So yes, it's within the scope of the proposal.
Okay. Thank you
for help clarifying.
Okay. So we don't have any other questions or raised hands for the moment. Oh, one has come in. I spoke too soon. Robert Cook asks, are there any thoughts as to which solute carriers or classes would form the shortlist for study?
I heard that. I just didn't hear the beginning. I'm sorry for the question.
Are there any thoughts as to which solute carriers or classes would form the shortlist for study?
Oh, so on the part two, again, this would be done collaboratively. So obviously, you know, coming from pharma, we have certain interests depending on certain diseases or their expression of human genetics, as well as those that might be involved in drug metabolism. But again, the intent is to define that short list collaboratively.
Can I just comment on that one, Claire? I think we've said that each company would be able to name a certain number of SLCs that they will want to study in detail. We haven't made that list yet, and I think we wanted to make it as part of the I think it's Work Package 4. Yeah, the companies would like to have the option to work with the academic consortiums to help refine that list. Some companies may already have a list already fixed that they, you know, we want these 10 or 12, whatever the number is. But other ones may want to work collaboratively to determine that list.
Okay, thank you. Okay, if anyone else has a question, I suggest you type fast because I don't see any for the moment. So as I said earlier, the core text is already online. We will also publish the recording of this webinar and the presentation itself. And if you have any questions after today, then do send them to us at info-imi.europa.eu. It's our job to help you. If it's a question about the core topic or a question about our rules and procedures, it doesn't matter. And if you keep an eye on the IMI website, then you will see as well when we launch the call. I would also encourage you to follow us on Twitter, join our LinkedIn group and sign up to our newsletter. Then you can be sure of always getting the latest information on all of our funding opportunities. I don't see any more questions, so I think we can close the webinar. So thank you very much to Claire for the presentation, and also to Hugh, and for all of the other FP writers who chipped in with writing the proposal, and of course especially to David for answering questions here on the day. I wish you all very good luck with your proposals, and wish you a nice day. Goodbye, everyone.
Bye. Thank you.
Bye.